The following was recorded in March 2019 at the Endocrine Society's annual meeting in New Orleans.
Hello, I'm Aaron Lohr, and this is the Endocrine News Podcast. Today we'll be talking about how time-restricted eating may help prevent breast cancer. With us right now is Dr. Manasi Das of the University of California, San Diego. Thank you for joining us today.
Thank you, Aaron, for inviting me to share my research. And currently, I'm working as a postdoc, as you mentioned, in the University of California, San Diego.
And we're very happy to have you with us. Your title of your endo presentation is Time-Restricted Feeding Delays Breast Cancer Initiation and Growth in a Mouse Model of Postmenopausal Obesity. So as your abstract title implies, your work involved a mouse study of postmenopausal obesity. But before we get to your results, I just want to talk a little bit about the association between postmenopausal obesity and breast cancer. What do we know about that sort of relationship?
As we know, breast cancer is the most common cause of cancer mortality in women in the developed country, and the second most common cause of cancer mortality in the developed countries, and the most common cause of cancer mortality in the developing countries. And obesity is really a high risk factor for breast cancer, and particularly in postmenopausal obese women. And in fact, if we can see the studies has been done, the chances of having breast cancer increase by 40% in the postmenopausal women compared to anything.
That's a very significant number.
Yeah, it's a very significant.
Now, when most people think about dietary interventions, they're thinking about what they eat, not so much when they eat. And your study is looking a little bit more at when we eat. So why is the when so important?
Yeah, it's very true. Like when people think about the dietary intervention, the first point they think about is what they eat. But like research suggests that like it is also important when we eat than like what we eat. And before understanding why this when is important, we should know like what are the factors that links obesity with the breast cancer. Studies suggest that like the circadian rhythm, that means like activity of a normal human being throughout a 24-hour of time, the normal metabolism, that those are the factors that actually links obesity with the breast cancer. And when we switch our timing of eating from a normal regular time to a non-eating time, like for example in the night time, the circadian rhythm or the metabolism, they gets deregulated. So if we do that, like we are having more chances of having breast cancer than if we don't follow that one. So it is really very important when we eat than what we eat. And if we take an example of like, for example, the Ramadan fasting events, that's a religion event when people do fasting in the daytime, they have very small amount of improvement in their metabolism and the circadian rhythm, but like because they are just switching their fasting time, instead of like nighttime fasting, they are just fasting in the daytime. That's why they are having less improvement in their metabolism. But like studies have shown that for prolonged nightly fasting condition, they have shown improved in the metabolism as well as like the biomarker for breast cancer risk. So they have shown that like prolonged nightly fasting is improving your biomarker for breast cancer. So that's why it is more important when you eat, not just what you eat.
I really appreciate that explanation. It's something that I care about. You know, I'm someone who's probably trying to lose weight, and I think about what hours are best to eat and which hours are best not to eat. So when we say like a nightly fasting is being a good idea, is it a stretch of time, like a 12-hour block, or?
Studies have shown that in human women, a minimum of like 13-hour fasting is needed if you want to take care of your breast cancer risk, like reduce the breast cancer risks. Mm-hmm. But even if 10 to like 12 hours of fasting is sufficient if you just want to lose your body weight or improve your metabolism. in the nighttime.
So I want to come back to your study because you have a mouse model of postmenopausal obesity. Can you describe how you developed that model?
Yeah, so this study actually has been done in the postmenopausal obese mice model. So we actually developed two kind of postmenopausal mouse model. In first mouse model, what we did, we just surgically removed the ovary of the mouse and then put them on the high-fat diet, like 60% high-fat diet. That's a very rich high-fat diet, but it is similar to what we are eating, the burger and pizza every day. So and that's the one model we developed. And in another model, we injected the mouse continuously for 50 days with a chemical called VCD. That's caused ovotoxicity. And that's how we are getting rid of like the ovary at the end of the time frame of injection, and then that eventually will develop the postmenopausal obese mice model.
And then at some point with the model, you had to introduce the development of breast cancer. So how did you bring that in?
So after actually like having the mouse model develop and then having them feeding on the high-fat diet to make them obese, like after they gain certain body weight, like 45 gram, for example, we consider them as obese. And then at that point, we injected 2 different breast cancer cell lines into the mammary fat tissue, sub-Q. So that's the area like where generally the breast cancer develops. So we injected them sub-Q with the 2 breast cancer cell line that is syngenic like to the mice BL6 we used.
And at any point, did you look at hyperinsulinemia?
Yeah, we definitely did. Actually, in our study, we studied various metabolic parameters, which includes the glucose sensitivity, insulin sensitivity, and then we saw that like with the time-restricted feeding condition, we are controlling the body weight, we are controlling the glucose sensitivity, and then we are also controlling the insulin sensitivity of the mice.
So now that we have a good understanding of the mouse model and how you developed it and what you did, what effect did you find that time-restricted eating had on tumor development and growth? What did you find? Like
most important factor of our study is like we found that like with the time-restricted feeding, we really had a significant reduction in the tumor growth and as well as in tumor initiation. That's really very significant, and we bring down the tumor growth up to the level to the tumor growth where the mice are on the normal chow condition basically, instead of they are on the high-fat diet. So the tumor are still comparable to the mice on the normal chow condition.
This is fascinating. Did you have any explanation as to why you would see this anti-tumor effect?
Yeah, sure. Like for example, because in our preliminary study, we study what is the effect of the TRF on the metabolism itself, and then there we found that the TRF is improving your insulin sensitivity. So we thought maybe insulin, because we know that insulin is a very potent mitogenic factor, so we thought that maybe time-restricted feeding is following insulin-dependent mechanism to reduce the tumor growth. So for that, like to understand that thing, what we did, we implanted insulin pump that will deliver a constant dose of insulin throughout the day in a normal chow feeding mice, and then injected the same breast cancer cell line to see first the effect of the insulin on those breast cancer cells on the tumor development. And we found that compared to the saline pump implanted mice group, the insulin pump implanted mice group is having higher tumor growth at the end of the study. So that gives us an idea that yeah, insulin is a driving factor there. And to further validate that thing, what we did, we took the obese mice and then again implanted the insulin pump into the mice on the high-fat diet. And then as a control, we had the saline pump group. And we found that even if, like with the obese high-fat diet mice group implanted with the saline pump, the tumor growth was reduced, but with the insulin pump group, the tumor was significantly higher. That's clearly indicate that definitely insulin is playing a more important role in driving the tumor growth here, and then our time restricted feeding intervention is acting through insulin dependent mechanism. And to further validate this thing, we took different tumor from different groups and studied the insulin signaling in the tumor itself, and then we found that the insulin signaling is reduced by the time restricted feeding intervention compared to the ad libitum high-fat group. That's how we come to a conclusion that insulin dependent mechanism is being taken care by the time restricted feeding intervention here.
After this wonderful explanation of your work, a lot of our listeners are probably wondering what's next. We know that this is a mouse study. Do you see this carrying over to people? Is this something, this time-restricted feeding and breast cancer risk, is this something that we as people should be considering as well?
Yeah, it's already being implemented in one of the study in the human patients, in one small cohort of the women with postmenopausal obesity condition. The prolonged nightly fasting, that's a time-restricted feeding intervention, has reduced the breast cancer marker. But I vision this is going to be a non-pharmacological approach to reduce breast cancer in near future.
Thank you so much for taking the time to share your work with us on the podcast today. I really appreciate it.
Thank you so much for inviting me here.
That's all for this episode. Thank you for listening to the Endocrine News Podcast. To learn more, visit www.endocrine.org/podcast. There you can find this episode and some helpful links. You can subscribe to Endocrine News Podcasts on iTunes or wherever you get your podcasts. Be sure to leave a review on iTunes. And if there's a topic you'd like to see us cover on the podcast, let us know by emailing us at podcast@endocrine.org. Thanks again for listening.
Endocrine News Podcasts are a free service of the Endocrine Society. To learn more or to become a member, visit the Society's website at www.endocrine.org.